Continuing on, we are continuing with Chapter 1, Part 2. We've gone over the basic structure of the cell and the general definitions of pathophysiology. Now let's see who is invited to the party, okay? And what I mean by that is who is coming in and out of the cell, because it is very important what comes in and out of the cell. We need to talk about how things get in and out of the cell. And the way that books define this and break this down is into passive and active. And what active means is it uses ATP. Now, ATP is created in the mitochondria. If you remember those little bean-like structures that are in cells, it's one of the organelles. And when we ingest glucose or foodstuffs to make glucose and we breathe oxygen, those little mitochondria makes tons of packets of energy known as ATP. Every A&P student goes over that. I don't know if you remember or not, but it's an important concept. And this ATP, through another series of complicated steps, helps to give our cells the energy all the cells need to function on a normal basis. Are there ways to make energy other than glucose and oxygen going into cells? Sure, it's not nearly as efficient, if you remember this or not. And of course, if you have a question, shoot me an email, okay? So the first grouping with this cellular transportation is passive. It doesn't use energy. The second grouping uses energy, okay? Now, when we're talking about cellular transportation, something that is super important is to understand solutions. Very simple. A solution is a solvent with a solute dissolved in it. So I always remembered solvent being the major liquid because the V looks like a W to me, and water is generally really good solvent, if that helps you to remember. Solute are the substances that are dissolved in the solvent, and they create a solution. And you guys know solutions, right? You could probably dance rings around me when it comes to solutions and all the solutions that you give to patients. So you need to know what's broken down in the solvent so you know you're giving that patient the right solution, okay? The solution inside of the cell and outside of the cell is in constant change. Cells are sending things in and out, and that ability to move things in and out of a cell helps to balance it and maintain homeostasis for that cell. So we need to have different ways to move substances into and out of the cell to create balance. Okay, so you need to understand the solution outside of the cell versus the inside of the cell and how we get things in and out. And how we get them in and out is either by passive or active means. And the active means uses the energy, uses little packets of ATP. Passive obviously doesn't, okay? Now, diffusion is a movement of solutes from high to low concentration. So if we have open channels in the cell, those protein membranes that I spoke about earlier, if they happen to be open, we could just sort of scoot things in and out. And with diffusion, things will move until they are balanced throughout the solution. I always think of diffusion being like a party. If you put 10 people in my house, we aren't all going to congregate in the living room, although everybody seems to end up in the kitchen, right? When you're hosting a party. But anyway, people would diffuse throughout the room and would balance the solute in the solution. So they're going to move from high to low concentration. Everybody isn't going to jam in the corner of the room. They're going to spread out, okay? Now, osmosis gets a little tricky. I don't know if you remember this picture or not. It's the movement of water or another solvent. We're always going to be talking about the water in the body if you just want to make note of that. From high solution to low, but it will move through a membrane, okay? It'll move through a membrane. I have a great picture of this I'm going to show you in a little bit. Okay, facilitated diffusion. Molecules are going to move from low to high concentration with the carrier molecule. Don't even worry about it. We're never going to talk about it. I hate to say that I put something in that we're not going to talk about. Don't worry about it. Worry about diffusion and osmosis. Okay. Now, with active transport, there's three options. Sodium-potassium pump is going to be one of those protein pumps that passes through the cell membrane. Okay. And what that helps to do is to reestablish the proper ion concentrations of sodium and potassium inside and outside of the cell. That's all. This is a complicated concept. We're going to get way more into this in the future. What I want you to know in your little brain is that I have in my cell membranes a little protein channel called the sodium-potassium pump that's going to balance sodium and potassium on the right sides of the cell. I'm not going to tell you what goes where because that's a whole different conversation for the future. Okay, so sodium-potassium pump is going to balance sodium and potassium ions. It's going to use ATP. It's a very important part, I don't know if you remember, of action potentials. And that's a whole different animal unto itself. Okay, so just for right now, wax on moment. Just memorize what I'm telling you. Sodium potassium pump uses energy, balances sodium potassium. Got it. That's all we'll talk about for now. Endo and exocytosis. I hate to sound crass, but it sucks things in or it spits things out. Endo means taking in, as you know, from an endoscope. Exo means spitting stuff out. So endocytosis will happen in processes such as phagocytosis. And phagocytosis happens with our immune cells to bring in substances that may be harmful or infectious for us. And it'll release enzymes that breaks it down and destroys it. Exocytosis, if we suppose we create a certain protein inside of a cell and we need to excrete it out, we'll excrete it out by exocytosis, okay? And all three of those use energy, okay? Simple diffusion, we have little fat particles. They can sneak right through the membrane of our cell. And you can see here we have the outside on the right and the inside on the left. If we need to get little fat particles in, they could just sneak right through that cell membrane. Oxygen can sneak through there as well, okay? They could sneak back out if they want to. Simple diffusion. And it's passive because it doesn't use energy. This is an example of osmosis. So now we're talking about not the movement of particles, we're talking about the movement of water. And this is important because water can go in and out of cells according to the amount of molecules that we have inside and outside of the cells. Water will go where there's more molecules. So if we're looking at this bag, they give us an example of a bag with sugar in it, okay? If we have more molecules on the inside of that bag, more water will move inside of the bag, as long as that bag is permeable to water. And I'm talking about the bag as if it were the cell membrane, okay? So if we have, you know, a ton of particles in here, and suppose this is a cell, water will start to move in because water can pass through that cell membrane, okay? On the other side, if we have a lot of particles out, I don't like this picture for this example. If we have a lot of particles out, we can move water out of the cell to the environment, okay? So water will go where the particles are. And the reason that's important is because if we have a lot of particles inside and we move a lot of water inside, I mean, think about how small that cell is. It'll expand that cell and it can break apart. If we move a lot of water out of the cell can shrink because we're moving water out to where the particles are because it's trying to balance the solution, will shrink that cell. So basically what to remember is that our cells are going to try to balance the solution outside and inside of it. And if there's a lot of particles inside, it'll bring more water inside to balance out that solution to be more similar to the solution outside of the cell. It can expand it. If we have a lot of particles outside of the cell, as opposed to inside of the cell, we'll move a lot of water out and we'll shrink the cell. Well, cells need to be a proper size in order to function, okay? So we want to keep a nice balance of solution inside and outside of the cells. It's a little hard to explain. So if this doesn't make sense, by all means, shoot me an email. Here's a picture of endo and exocytosis. Okay, we either suck it in or we spit it out. And that's just about it. And it uses energy. That's a good thing to remember as well. Okay, sodium-potassium pump. Like I said, this is a complicated thing. It's and we're going to get into it when we talk about action potentials in the future. Basically, I think the gist that I want you to know now is, in a resting cell, this is a good start. In a resting cell, we are going to have more sodium on the outside. This is a resting cell. Cell is just hanging out, okay? And sodium is represented here by Na. Sorry, this is so sloppy. It's hard to write with us. Na+, okay? Sorry. I don't know why when I'm writing it does this sometimes. Okay, Na+, and you could see that there. Now, potassium in a resting cell is going to be on the inside of the cell, okay? That's just how a resting cell is. So if we're looking at a little resting cell, you're going to have more sodium on the outside, more potassium on the inside, and you can see it's represented there with K+ to the right. Okay? All right. What the sodium-potassium pump does is if it senses that the solutions are getting imbalanced, it's going to pump more sodium out to reestablish more sodium on the outside of the cell, and it's going to pump more potassium in to reestablish more potassium on the inside of a resting cell and reestablish that resting cell membrane. It is way more complicated than that. I want you to understand just that basic, wax on, wax off, just understand this a little bit, okay? So the sodium-potassium pump, what I want you to take away from that, and if you understand 20% of this, this is all we need for right now, is it is active, it uses energy, it's going to pump sodium out of the cell and potassium into the cell, and that's all we need to know for right now. That's it, okay? And we're going to get way more into it in the future. And it has to go through that little protein channel. And this is a great picture showing you that protein channel. Question time. Active transport includes which of the following? Diffusion, osmosis, sodium-potassium pump, endocytosis, 2 of the above. Write down your question and check to see if you're right. Cell cycle. Okay, moving on. So we've gone over the basic structure of this cell, how we get things in and out, the premise that we need to keep cells balanced with our solutions inside and outside of the cells, or the proper shape cannot be maintained. Now we're going to talk about how those cells replicate and how they react to damage. Cells replicate through mitosis, okay? And with the normal cells, and I spoke about sloughing off skin cells, we got to create more skin cells. When we create more skin cells, we're going to create cells with 23 pairs of chromosomes, so a total of 46 chromosomes inside of that cell nucleus. So if you can picture the cell itself and then the nucleus in the center, inside of that nucleus we will have 23 pairs of chromosomes. They actually dissociate and form this kind of just bowl of spaghetti of DNA, but don't let the blue run. Sorry, don't let the blue run. But we'll have 46 chromosomes. So 23 pairs, 46 total. When we have cells that undergo meiosis, this will happen with sperm and oocytes, which are known as the eggs in the female ovary. They undergo a process called meiosis, which causes or creates half the chromosome number. Okay, and we do that so that way when they get together, they form 46, which is the 23 pairs again, to create a normal number. If we don't have a normal number of chromosomes, we'll either have some sort of congenital issue or abnormality, or it doesn't create a viable baby. So it's very important to have the proper number of chromosomes, and that's the job of meiosis. And I didn't create, I wish I could. I copied and pasted this picture from your book that shows mitosis versus meiosis. We're not going to get into the specifics of this. I like it because it shows all the different steps. What you need to know is mitosis will create 2 daughter cells containing the 46 chromosomes, okay? And a cell that has the full complement of 46 chromosomes are known as a diploid cell. And that's right at the bottom there, right here, okay? So each of these daughter cells contain the proper amount of chromosomes. With meiosis, it goes through several steps. Okay, and I'm not going to ask you about the steps, but I want you to know that it's going to create a haploid cell. And I always remembered haploid because it sounds like half the chromosome number. This only has 23 chromosomes, okay, not the 46. So that way, when an egg and a sperm meet, it'll create that full complement of 46 chromosomes, and we will hopefully have a happy, healthy baby. Okay. Cell differentiation will happen when a oocyte is fertilized by a sperm. And as we all know, I love this story, we don't start as little people, right? There was actually a scientist who believed that a man ejaculated little people, and those little people got embedded inside the uterus. It's crazy. And then grew. That's not how it happens. We know that. What happens is the egg and the sperm meet. They form a cell with a full complement of those 46 chromosomes. The cell divides, and it divides and divides and divides. We're not going to go into specificities of that at this point. But that division and all the steps it goes through is called cell differentiation. And the cell divides, then it forms layers, and then from those 3 layers come all the organs of the body. That's a very complicated process in and of itself, so I'm not going to go into those specifics, okay, at this point. But cell differentiation, excuse me, it's fascinating. Every time I was pregnant, it blew my mind that there were so many crazy steps. Question time. The main difference between mitosis and meiosis is that cells have 46 pairs of chromosomes after mitosis and 23 pairs of chromosomes after meiosis. I want you to read every word before you answer. Cell changes. What do they do when they're irritated or ignored? What do you do when you're irritated or ignored? All right. What do we all do, right? Here's some great pictures of and there's some great pictures coming up. There's some great descriptions here of what happens to cell changes, and these are very basic pathologies that can happen to many of the cells of the body. Atrophy. Great example with atrophy is somebody who has had a cast on their arm or their leg, and you take it off and you're like, wow, their leg is so much smaller. Well, there's been a decreased workload on that extremity, which decrease the size of the organelles, or the teeny tiny organs in that cell. They have a decreased energy usage, they shrink, and they have a decrease in function. Well, in this case, once you get the cast off, you can exercise and get it back to normal. Okay, so you can help to fix these cells. This is not irreversible. In some cases it is, but in muscular cases it's not. Hypertrophy. So you have an increased workload. Now, this could be normal, such as an increase in muscle size if we work out and lift weights, but this can also be with disease. And I think a great example of this is the heart. When the heart undergoes abnormalities, for example, if we have atherosclerosis, or the hardening of the aorta, the heart has to work harder to push blood out. It'll hypertrophy. You would think that it would make it stronger, but it actually makes it a little flabbier because it stretches out the cardiac cells, and they can't contract as well as they would if they were a normal size. Okay, so hypertrophy could be normal or abnormal. Increased size, increased number of organelles, increased contractility, unless you have a disease state such as a flabby heart from severe hypertrophy. Okay, and that can decrease the functionality of the heart itself. Hyperplasia is an increased workload. Can also be disease, such as his hyperplasia of the cervix. So you have an increased tissue size due to the increased number of cells. So being very specific with those definitions, hypertrophy is an increase in the size of the cell itself. Hyperplasia is an increase in the number of cells. So you're going to have, the cell itself doesn't grow. The number of cells will replicate quicker. Okay. An example, when I was in chiropractic school, I'm actually a chiropractor by trade. I took so many notes. I got hyperplasia of my pinky finger because I would push it, and I was so stressed. I would push my hand down to take notes, and the outside of my pinky finger, right at the joint where I would push it down, had hyperplasia. There was an excess amount of cells there from all the stress, from all the pressure. And once I stopped taking notes, guess what? It went away. Okay, so that's a good example of that. Excuse me. You can also have this with hormonal as a good example that your book gives with hyperplasia. The sloughing off of the endometrium, okay, the inner lining of the uterus. We will build it up, we'll increase the cell number, and then we'll slough it off. Okay, so that's the hormonal. So compensatory would be the rubbing of the finger, taking the notes, as you are hopefully taking frantic notes right now. No, I hope you're not. I hope giving you the notes makes it easier. And hormonal will rebuild up the inner lining of the uterus for menses. Metaplasia, here is the key word. Normal cells are replaced by abnormal cells. Okay, normal cells are replaced by abnormal cells. So one example is Barrett's esophagus. So with Barrett's esophagus, I don't know if you understand this, but in the stomach, we have a certain size and shape of cell. It's actually called columnar. It's tall and thin. And in the esophagus, we don't. We have kind of these egg-shaped cells in the esophagus. And there's a very, very quick differentiation between the two. I'm going to try to actually look for a video for that for you. So what happens when people have reflux is the acidity of the stomach moves up into the esophagus, and you actually change the cells in the bottom of the esophagus to more of that columnar shape like it is in the stomach. So that's an abnormality, okay? And cigarette smoking can change the cells that line the trachea and the bronchi, okay? So very bad. You actually lose the cilia. So that becomes a pathology. And what the problem is with that is in the cilia can't clear junk out of our lungs as well, because that's part of the job of the cilia in the respiratory tract. Dysplasia. Excuse me. So normal cells become an abnormal shape and size. So we can see this with epithelial tissues. So I think the most common one that we'll see this is precancerous changes in the cervix. Okay, so not only can we create the number or have hyperplasia within those cells, we can also have a mutation of the cells to an abnormal shape and size. Okay, quite often it could be dysplastic, could be precancerous. It doesn't always have to be precancerous. Okay, so abnormal shape and size. I feel like with these cells, there's a little crossover with them. So I will keep the questions, as I said before, directly from what I've given you in these notes. Okay, so for example, dysplastic is going to go from normal cells to abnormal shape and size. Metaplasia is going to be the replacement of normal cells with abnormal cells, etc. To make that more straightforward. I feel like clinically, people kind of interchange some of these things, okay? And that's okay, but I'll try to be as straightforward as possible. And this is a great, picture of all these cells. I would have to, of course, if I was showing you hypertrophy or hyperplasia, give you normal cells to compare it to. Okay, metaplasia versus neoplasia. And we didn't really go over neoplasia. That's going to be a whole separate thing. I meant dysplasia. Okay, so metaplasia, the replacement of those normal cells with abnormal cells. Dysplasia, they're trying to show you the change in the size and the shape, and atrophy, of course, is going to be the decrease in that size. So once again, I'll take it right from those notes because I do think it kind of gets a little wonky here with a crossover. Neoplasia, we'll talk about it about its own separate entity. Okay. Question time. When cilia are lost in the respiratory tract, this is an example of what type of cell change. This is a little difficult, this question. Think about the true definitions of what I have given you. Okay, so cilia are lost. We have a cell with cilia as opposed to a cell without. So would that be metaplasia, dysplasia, hyperplasia, or hypertrophy? I'm going to pause the tape at this point, and I will see you in the next video.